Hello, my name is Izik Engelberg, and I'm really proud to be here. I'll take this stage to first thank the organizers, which gave me the opportunity to speak here. It's a great privilege, so thank you. I'm about to talk about antimicrobial peptides and their property of forming functional suprahelical assemblies. And together with my supervisor, Professor Meitalando, we're about to show you a very, very interesting and promising study. Antimicrobial peptides, or AMPs, are secreted by most organisms on this planet. AMPs kill bacteria, they kill viruses, they kill fungi, but moreover, they're actually used as the first line of defense of the host against pathogens. The abundance of AMPs in the material science is pretty surprising, and in a very nice study being done by Teixeira in 2020, you can see that the most quoted searches for AMPs in the material science are in the multidisciplinary field and in the nanotechnology and nanoscience field. AMPs in the material science, the most time which you will find AMPs in the material science, you will find it probably conjugated into another chemical compound or either encapsulated inside a polymer. But this is not the only option where we can use AMPs as a material. AMP is known to form fibrils, at least some of them, and those fibrils are active. They have a purpose and they have meaning. So if they have a purpose and meaning and action, we can use them. For example, you can see here that they can be used as a sole and independent delivery system. They can entrap bacteria, they can modulate the immune system, and more. In this talk, I will focus on human-derived AMP. His name is LL37. LL37 is one of the most common and studied AMPs from humans. In my study, I focuses on one of the segments of LL37 between the residues of 17 and 29, and you can see the atomic structure which we solved in pretty high resolution at 1.5 angstrom here in the right side of the screen, and you can see it from the side view and also from its top. And if you take a look at the top view of the helix of this peptide, you can see very interesting phenomena. All the charged residue, all the polar residues goes into one side of the peptide, and all the non-polar residues goes to the other side of the polar. And here I lined these polars and I indicated for you. And this is a very unique but common phenomena which characterized membrane-active AMPs. Remember that we will use it later on. Besides being a very interesting and very unique peptide, LL37 17 to 29 also assemble into a tetramer. This assembly is, as you can see on the right side of the screen, assemble under the forces of hydrophobicity. All the non-polar residues, which you saw before on the downside of the peptide, comes together and form the hydrophobic core, here colored by red. You can see on the center of the screen that if we will annotate due to charge the atomic structure of the tetramer, we can see a very positively charged particle, giving sense to the assembly and giving sense to the stability of this particle. So LL37 17 to 29 is a very unique peptide. We also saw this peptide assemble into tetramers, in which those tetramers are highly cationic, highly charged. And now I can show you that according to the atomic structure, these tetramers are also further assembled into a higher hierarchy architecture, And you can see on the right side of the screen that these tetramers assemble into a fibril. And you can see that if we color this fibril due to the annotation of hydrophobicity and polarity, you can see a zigzag belts of hydrophobicity and polarity. And this is, of course, giving sense to this fibril to be an active species against bacteria. You can see on the left side of the screen the performing of a negative stain temp and performing a native cryo-EM present us that this peptide assemble into fibrils also in terms of nanometric scales. In order to emphasize the assembly of LL37, 17 to 29 into tetramers and the assembly of those tetramers into fibrils, we present here the atomic structure of this peptide. And you can see here, first of all, that a tetramer of these chains is colored here with pink. And if I play this movie, you can also see the presentation of the amino acids on these chains. And you can see that the polar residues turning outside and the aliphatic residues turning inside, forming a core. And you can see the position of this tetramer in the fibril, and you can see the repetition of these tetramers inside the fibril as I rotate it. Okay. Besides being such a visual fibrils, these fibrils of this AMP are very thermostable. We previously saw that according to the atomic structure, the assembly of the tetramers is designated to be highly biochemical stable. It's formed due to the hydrophobic core. It exposes all the polar residues and all the charged residues outside and is highly cationic, highly charged, very stable in a solution. How stable in a solution? You can see at the downside of the screen that its zeta potential is even higher than collagen. And collagen is one of the most biological fibrils in terms of solution stability. In the upper side of the screen, you can see that these fibrils can bear the heat of more than 80 degrees. Next, we wanted to correlate between the atomic structure and the self-assembly of the tetramers to their antibiotic activity. You can see on the left side of the screen the atomic structure of the tetramer, and you can also see different clouds with different colors indicating different residues. For example, purple and orange clouds indicating lysine and glutamine residues. And you can see on the right side of the screen, it is antibiotic activity chart indicating the growth of the bacteria exposed to these peptides. And you can see, for example, that the y-axis indicating the growth rate of the bacteria. And for example, that the gray bar here of the LL37, 17 to 29, the native peptides, indicating this peptide to be very efficient in inhibiting the bacteria growth. But you can also see that the purple bar here and the orange bar here are mutations of the residues indicated on the left side. And you can see that these mutated peptides were not abolished in terms of antibiotic activity. But on the other hand, you can see on the left side again that green and blue labeled residues, hydrophobic residues such as isoleucine and phenylalanine, and going back to the right side of the screen, while mutate these residues into inert residues such as alanine, abolish completely the activity of this mutated peptide. So this allows us to correlate between the necessity of this self-assembly into tetramer to the antibiotic activity of these peptides. To conclude this part, we saw that LS37 17 to 29 forms very unique superhelical structures with zigzagged polar and hydrophobic belts. We saw that these fibrils are functional. The assembly into fibril is essential to the antibiotic activity. And we also saw that they are thermostable. They resist to heating. We also determined the localization of these fibrils on the bacterial cells, as you can see on the right side of the screen, and you can see the fibril of the peptide merging out from the membrane of the cell. The next chapter I want to talk about is to select something nature chose to deselect. How can we determine that something undergo extinction? How can we determine why nature chose to extinct such compounds? We're going back to LS37, 17 to 29, and those figures you saw before. On the upper side of the screen, the atomic structure from the top view of the helix, and on the bottom side of the left side of the screen, you can see the tetrameric assembly formed due to the hydrophobic core. We wanted to ask ourselves what would happen if we will take this hydrophobic core and we will covalently bound it. We replace the most hydrophobic residue inside this core. This is isoleucine into a cysteine residue. And we know that cysteine form disulfide bonds, covalently bonds. So we expected this tetramer to be covalently bounded. But what we get is very different. Surprisingly, we saw that the peptides are very similar to LS37, 17 to 29 in terms of helicalicity, and the residues turning for each side. The polars goes up and the hydrophobic goes down, and you can see that the cysteine and the disulfide bonds are at the right position and they form intermolecular interaction. But what we further found that the assembly of the peptide formed by these mutations of cysteine are very different from what we saw before for the native LS37, 17 to 29. You can see on the upper side of the screen, again, the atomic structure of this mutant. And you can see in the fibril atomic structure that this mutant is stabilized by the formation of the cysteine disulfide bonds, here colored and indicated with yellow. And you can see repetitively the intermolecular interaction by the cysteine. But you can also see that this fibril is again stabilized also by hydrophobic core. And you can see it in the center of the screen. And you can see that annotating this fibril due to hydrophobicity and due to polarity indicate us that the polar residues of these fibrils turning outside and the hydrophobic residues turning inside, forming a hydrophobic core elongated along the fibril axis. You can also see on the downside of the screen that amazingly, these fibrils are super cationic charge. They're completely blue here with the annotation of the positive charge. This gives us sense for the assembly and the thermal. This gives us sense for the assembly and the thermodynamic, pushing these peptides to form higher hierarchy assemblies. In order to find what is the role of the disulfide bonds in the activity and the formation of structures by these peptides, we simply added the reducing agents. The reducing agent, here indicated by DTT, simply supposed to deform the disulfide bonds. And what you can see on the left side of the screen, that first, the addition of DTT to the bacteria tested here is not harming them, not by means of morphology and not by means of toxicity. But what you can also see here on the downside of the screen, at the left side of it, that the addition of I24C, the cysteine mutant inside the hydrophobic core, forms very dense structures on the bacteria membranes. You can also see that a bit right from this image, while the addition of DTT, the defined structure we saw before, is now diffused, and now we get a very non-structures assemble. And this is also backed up again with antibiotic assays. You can see on the right side of the screen that first of all, on the left side, the addition of DTT to the native peptide, which does not contain cysteine residues and therefore does not contain disulfide bonds, is not affecting anything. There is a certain level of antibiotic activity due to the peptide concentration, and at some points, if we will reduce the concentration of the peptide, the bacteria will happily ever after grow, and this is not affected by DTT, as you can see here. But you can see that while adding DTT to I24C, suddenly at some point we get the abolition of activity, the addition of reducing agent causing the first deformation of the structures, as you can see on the left side, but it's also caused the abolition of the antibiotic activity. So we can definitely correlate between the necessity of the assembly of these peptides into their antibiotic activity. So in the beginning of this talk, I described something we named here as a phenomena which nature chose to deselect, and why we present it this way. Because you can see here the work of Peleg from our group, and Peleg found that the cysteine frequency at general AMPs, you can see here at the left side of the left chart, is pretty prevalent. Cysteine is prevalent in AMPs, period. And you can see that as a control, tyrosine residues and arginine residues are also prevalent in somewhat differences from cysteine. But you can see on the right side of the bars of the left chart, these bars, that the prevalence of odd number of cysteine residues in AMPs is dramatically decreased. Odd number of cysteine residues obligate the chain to form intermolecular assemblies. So nature devoid of forming these intermolecular chains formed by disulfide bridges. That they are tautonability. So to conclude the second part of this talk, we saw due to the statistic work of Peleg that nature devoid of using odd-numbered cysteine residues inside amphipathic helical helixes. These are antibiotic peptides which are membrane active. The reason for that we believe to be the high tunability of these residues. So simple addition of reducing or oxidative environment can alternate the activity of these peptides, and nature devoid of using such alternated compounds. But we humans know that we can use this fact. So cysteine residues can easily be used as a switch. I will summarize this talk. The antibiotic activity of AMPs is crucially correlated to their self-assembly. And on the other hand, the self-assembly can play a crucial role on the antibiotic activity. Of course, we humans like to use this in order to design novel compounds which can replace the collapsing system of antibiotic molecules, which are known to lose their activity against resilient strains of pathogens. These novel compounds of AMPs, tunable AMPs, can be used in many biomedical approaches, such as dental using tissue engineering, scaffolding, smart tissue patches, medical devices coaters, and more. I would like to thank you all for listening, and I would like to thank to my supervisor, Professor Meital Landau, and to my beloved group, which suffers me every day. And thank you all again.